Hello friends, this week I had posted one question. Let's discuss the question and figure out what's the answer. The question goes by the title, Who am I? So the hints are given, clues are given, and on the basis of those clues you are expected to identify this. So the first clue was, I need two neurotransmitters for my activation. Now since it's a neurotransmitter which is mentioned here, that means it must be receptor, but it requires two neurotransmitters for its full activation. Then second clue is, I am voltage gated as well as ligand gated. Yes, the channels are either voltage gated or they are ligand gated, but this is both. So again, very unique. And I allow the flux of two ions. Yes, it allows influx of sodium and calcium. So it requires everything in twos. So what is this particular, whether it's structure or a chemical or receptor, whatever it is, let's find out what this fellow or who this fellow is. The answer is, it is NMDA receptor for glutamate. You know, the glutamate is an excitatory transmitter, and more than 90% transmission in the brain or in the CNS is carried out by the glutamate. Now the glutamate has various types of receptors, AMPA receptor, NMDA receptor and kainate receptor. So these three are main types of the glutamate receptor. Now in this particular case we are talking about the NMDA receptor for glutamate. Yes, that was the answer. N-methyl D-aspartate, that's NMDA receptor. Now I require two neurotransmitters for my activation. That was the first clue. So yes, NMDA receptor, which is situated on the postsynaptic membranes, glutamate will bind to one part of this receptor, but it also requires glycine binding on the other part. You know, glycine is an inhibitory neurotransmitter, generally speaking. But in this case, glutamate action will occur only if glycine binds to the other site, other binding site on the receptor. So in that sense, two neurotransmitters are required for its full activation. Point number two, it is voltage gated as well as ligand gated. That was the point number two. So even when glutamate and glycine is bound, so ligand binding has occurred, yet the channel is not completely open because there is a magnesium block on the inner aspect of this channel, magnesium block, and this magnesium block will be removed or can be removed only when the membrane is partially depolarized. So when the glutamate binds with a receptor, there would be a partial depolarization of the postsynaptic membrane, and that would remove the magnesium block on the inner aspect of the channel, and then the influx of ions can occur. So therefore, voltage gated as well as ligand gated. Ligands, the glutamate and the glycine, and there was a partial depolarization which was necessary for complete opening of this channel. So again, voltage gated also comes into picture. And the third thing is that when this magnesium block is removed and the channel is completely open, then it allows the influx of sodium as well as calcium. Though the calcium influx predominates, it's the major event. However, it has been found that this channel is permeable to sodium as well as calcium. So again, flux of two ions, again unique, because normally we have the channels which either allow sodium influx or potassium exit or chloride movement, they are individual channels. This is allowing the flux of two ions. So it requires everything in twos, and therefore very unique in that sense. The answer was NMDA receptor for glutamate. Now what is the clinical application of all of this? NMDA receptor for glutamate has been implicated in a variety of processes. For instance, long-term potentiation in hippocampus. You know, the hippocampus is the basis for long-term memory. And what's the basis for long-term memory in the hippocampus? Long-term potentiation. This is possible because of the NMDA receptors in the hippocampus. Glutamate transmission and action on the NMDA receptor is responsible for a long-term influx of calcium, long-term EPSP generation in the postsynaptic membranes or postsynaptic neurons in the hippocampus, and that's the basis for long-term memory. Then it's also been implicated in excitotoxicity to the brain. Penumbra, which is also sometimes called as the excitotoxicity to the brain, is because of the excessive glutamate action on the postsynaptic neurons in some part of the brain or ischemic area of the brain, resulting in long-term influx of calcium which damages the surrounding neurons. That's a matter of another video. And it's also involved in ketamine and its effect on dissociative anesthesia. Even there, this receptor has been implicated. So all in all, a very important receptor. Still research is going on and they are finding out new facets, new properties of this receptor. So in short, this was the answer to the question that I had put up on my social media platforms this week.